Is the recommendation to eat less protein a good idea?
It's very vague in these days because protein is, of course, made up of different amino acids. Now you have specialists who are looking at the bark and the leaves and the roots and not even realizing that they're looking at a tree. That's a case of getting less and less about more and more until you know nothing about everything. If we just add glycine to the diet, we found it made the animals live longer.
One of the big mistakes here is looking at protein as protein and not looking at amino acids as very individual, not only building blocks, but signaling molecules.
One thing I would say that really kind of rubs me the wrong way about it is if you decide that something is non-essential and it turns out to be essential, you kind of miss the whole ball game then, right? You got nothing left.
What is your major discovery around glycine?
It's all about the glycine, because glycine is like a trigger lock on these cells that cause inflammation. Major discovery, except I already discovered it in 2010.
You're not saying plant proteins or that veganism is better.
That's because when you compare plant protein to animal protein, you're not including. What I found out was that glycine is the most extraordinary compound.
Hey everyone, there's a paper pretty much going viral right now that's telling people if they eat less protein, they might live a longer life. It's based on 300 studies. And fortunately, I have a scientist here, Dr. Joel Brind, one of your favorites, who has studied amino acids for decades and believes that we're looking at some great data and perhaps drawing the wrong or maybe an incomplete conclusion that there might be more to the story. So today we're going to talk about, are low protein diets a good idea if you want to live longer? Who might they be a good idea for, if they are even a good idea? And what might be other mechanisms or factors at play that are not being talked about in the mainstream? So thank you for coming, Dr. Brind. There's a paper, like I said, going viral right now telling people to eat less protein to live longer. You've studied this for a long time. So what do you think about this? Is the recommendation to eat less protein a good idea?
It's very vague in these days because protein is, of course, made up of different amino acids. There's 20 different amino acids, for the most part, that your body uses to make protein, all these different little Lego blocks. Proteins are the big molecules that they make stuff out of, like the fibers that support your skin and your bones, and the enzymes that convert one thing into another, and the carrier proteins and the enzymes, and all these are little machines, structural components, everything made out of protein. But protein's very heterogeneous depending on what amino acids are involved. And in addition, a lot of the amino acids have particular roles in the body in addition to there being building blocks of protein. And so to just say more protein, less protein is very vague. Why do they have scientific data going back? Because that's really a stand-in for how much meat you eat. Because the highest protein foods are meats, meat, fish, and poultry. So people who don't eat meat, fish, and poultry have low protein diets. People who do eat meat, fish, and poultry have high protein diets. And so that's kind of a surrogate for which amino acids you are eating. And so the idea of more or less protein is just way too vague to draw any scientific conclusions from. There are lots of people who eat high protein diets who do just fine. Lots of people who eat low protein diets who do just fine. Especially when we start getting into different amino acid composition and supplements and, all of that. That's really kind of vague. Now, in fairness to the authors of the article, I think the primary author, his name is Lamming. He's a professor at University of Wisconsin. And he's actually, like I was, in fact, looking at particular proteins. In particular, he was looking at methionine and leucine, the branched-chain amino acids. Those are the amino acids that are particularly rich in meat, fish, and poultry. Now, when I say that, I mean what we usually consider meat, fish, and poultry that we eat, which is the muscle meat. And so even to say, as many studies have shown over the decades, it's better for you if you're a vegan because not only are you eating less protein, but it's plant protein. It's not animal protein. Plant protein, well, the same amino acids are in plants and in animals, obviously, because if you didn't have the same amino acids in plant and animals, you couldn't be a vegan, you'd be dead. You would need to eat animal amino acids, right? Animal proteins. But you don't. So you can be fine and be a vegan. The thing is, animal proteins, when they say people on an animal protein diet and they study their diets as part of a whole nutritional study that they then publish and get data from and all of that. They're really not talking about the proteins in animals that you don't eat. In other words, what's in the bones and connective tissue, which is the stuff that your grandmother didn't throw away. She threw it in the soup, but we throw it in the trash for the most part, unless you're into bone broth, right, which we've talked about before. So it's what amino acids are you eating. You know, you can have a high protein and a high meat diet as long as you replace especially the glycine, which is very important. That's stripped, depleted actually by a high protein muscle meat diet, and so there you have these differences. So you have to look at the specifics. Now, to Lamming's credit, he's done a lot of work on the specifics of looking at methionine and leucine and isoleucine and what they do and all the different biochemical pathways that they have, that they play a part in. But he's in that regard, he's all into the biochemistry, you know, and they're not really talking about the important things, which is, in terms of glycine, it has to do with some of its non-biochemical roles, in particular its role in inflammation in the immune system. So a lot of times you get people who are, as I say, one of the problems with modern science is it's so hyper-specialized that even when I was younger, 30 years ago, I used to say that people were so specialized that you could be working in the forest and studying different aspects of different species of trees and not even realize you were living in a forest and looking at a forest. Now you have specialists who are looking at the bark and the leaves and the roots and not even realizing that they're looking at a tree.
Yeah.
So it's sort of a holistic approach tends to be more and more absent. We used to joke about that in graduate school and say as you go on in higher education you learn more and more about less and less until you know everything about nothing. So,
yeah.
To the large extent that's true. You have to keep the whole big picture in mind. Even a discussion about should there be more protein or less protein is kind of off the mark. And in particular, they've missed this non-biochemical role of this so-called non-essential amino acids. See, now this review by, what was the first author's name, and then Lamming is the...
It's Lamming and it's Bailey Knopf. Maybe?
Knopf and Lamming, yeah. So Lamming is the guy, the senior author and everything. It's a narrative review paper, which means it's not an original study, so it doesn't show any, original data. But it's not like Lamming is an outsider. He's got plenty of original data in this field. It's not like he doesn't know anything, but in the purpose of a review, a narrative review, is you give people a little piece. You take a whole field like protein in the diet, and you get a little bit about this amino acid, the essential amino acids, the non-essential amino acids, and on and on and on. And you summarize several hundred papers, 300 and some papers that have looked at these, and you try to give some kind of compendium or encyclopedia of knowledge about protein in the diet. And unfortunately, that's a case of getting less and less about more and more until you know nothing about everything. That's sort of the opposite of what we were just talking about. In fairness to this, I've written narrative reviews myself. My book, The Glycine Miracle, is largely a narrative review, but it focuses not on just one amino acid or on just one type of role that this molecule plays in the body, or one type of food that it's in, or one type of research study that's looked at it. It looks at everything related to it in regard to a very specific hypothesis that I have about the role of glycine in the body in terms of inflammation. So it's very general. So this review that came out is very useful. It's like an encyclopedia, except the one thing I would say that really kind of rubs me the wrong way about it is he talks about all the amino acids, including the non-essential amino acids, quote unquote, because that includes glycine. And he kind of gives them short shrift, kind of says, well, there may be something to it, but, because the body can always make as much as it needs, they're really non-essential. It's got lots of pathways to make glycine and all the others. So he really... You could see in his own mind he kind of, downgrades or downplays the importance or potential importance of the, quote, non-essential amino acids. So once you have gotten by that point of deciding whether something is essential or not, you're going to miss a whole lot. If you decide that something is non-essential and it turns out to be essential, you kind of miss the whole ball game then, right? You
Yeah.
you can't,
It's a big one.
you got nothing left because you've missed one of the key fundamental points, and that's true really of the whole field, not just lambing. So in fairness, he does acknowledge that there may be some merit to this, but really his focus is on like the diets which restrict the key amino acids like methionine and leucine and glycine. Plus, there are a few oddballs that are used very rarely, like selenocysteine and things like that. But they're all different, and they have different effects. So he's interested in looking at diets which are restrictive, and he does spend a lot of time talking about the science behind amino acid restriction, particular restrictions of particular amino acids, in particular, in this case, my own knowledge and experience, methionine restriction. And it was my work in methionine restriction, actually, which got me into glycine.
Tell us about that. Can you talk about those pathways and mTOR and FGF21 that they're proposing?
Oh, the FGF21 and the mTOR, those have to do with more specific actions inside the cell. The pathways that I'm interested in are really have to do with the nutritional biochemistry aspect, which is to say what these compounds are interchanged and interconverted into, rather than then finally what the signaling pathways are for them to, stimulate this or that, growth, inhibition of growth, that kind of thing. So that really, when you talk about mTOR and so on, metabolic stuff like that, those are signaling pathways. And that may have to do with the mechanism by which those signaling pathways are affected to produce something like life extension from methionine restriction. Now, restriction of methionine so I'll back up a little bit and then go back to, then we can get to where we are today. This goes back to the 1990s, as this review says. That's all in the review about how, the first paper showing methionine restriction was in 1993 by the Orenreich group. I was in that group. I was not in the group of authors because I wasn't working on that project at the time in the 90s. And what they found was that if you we were using Fisher 344 rats at the time, a standard laboratory model. If you restricted their diet in the content of methionine, which is one of the essential amino acids, and gave them, they could eat as much as they want, but the content of the diet was so limited that they would, no matter how much they ate, they would barely get enough methionine to survive. Yet, they lived 30 to 40% longer. Interestingly, they did not grow to full adult size. They seem to be happy. They seem to be, normal, not because you can make rats and mice, experimental animals, you can get them to be neurotic with the wrong diets and other treatments. And they were not neurotic. They seemed to be happy animals. They were even fertile, but they did not grow to normal adult size. But they lived 30 or 40% longer. So obviously this has to do with various signaling molecules having to do with growth and longevity and the intricacies of what makes a body age in that way. Now, the reason why that works, so over the years, methionine restriction was proven to be a real thing. First, the Orenreich group reproduced the finding a number of times, reproduced it then in mice as well as rats. Other laboratories involved in aging research also reproduced the findings in rats and mice. So it became a thing, right? Methionine restriction became a thing in the modern parlance, of aging and nutritional research. As a consulting biochemist, actually, to the Orenreich group, that wasn't my full-time job. I was a professor at the City University full-time. But I was a consulting biochemist at the Orenreich group, and they asked me to figure out why this happens. Why is it that methionine restriction extends lifespan? Well, I found out one of the reasons it does, because it ends up depleting glycine. And the reason I got into glycine was, what I was looking to do was, in particular, the hypothesis was, can we find something which will help the body to get rid of methionine from the dietary intake so that it mimics the actual effect of taking in less methionine? Now, the reason for that was we had tried and were actually publishing, there were a number of things published and still are. In fact, Lamming, I think, is actually, he's got a patent on a methionine-restricted diet. So he's been working on this, that's all I'm saying. And the trouble with that is that methionine-free diets or methionine-restricted diets are really very tasteless and boring and hard to stick to.
Tell us what the methionine-rich foods are. Just all meat.
Oh, outside of meat, fish, and poultry, all of that kind of stuff, things like mushrooms and lima beans and cheeses and dairy, all of that stuff is loaded with methionine. And there's almost nothing that tastes good. That's really. I proposed at one point in the group with Dr. Orenreich when we were working on this, because he wanted to have a diet that was methionine restricted. So I proposed a name for the diet. I said, why don't we call it the cream in your coffee diet? Because cream has less protein than milk, especially than skim milk. Those are very high protein. So if you like coffee, which doesn't have any methionine, and you want creamer in it, if you put mostly fat and less protein, so why don't you make it sound good by calling it the cream in your coffee diet?
I like it.
That might make it a little more palatable, but it really didn't go anywhere because it's very, very, very difficult to get people to stick to a diet like that. And if you're basically happy and healthy and you have the prospect of, maybe I can make it till 95 and not only 85 or something like that. Why do I want to restrict what I can eat? And, so my approach, and this was Dr. Orenreich's approach also, was to, can we find something which we can put in the diet which will basically like neutralize most of the methionine? One of the things that I thought of was a drug that is safe in hundreds of milligram quantities. And it's eliminated by the liver by methylating it. Now, in order to methylate something, which is to stick a one-carbon group on it, and to help to clear it in this particular case, it takes an S-adenosylmethionine, SAMe, which is activated methionine. So if you use up a lot of methionine, a lot of SAMe, you're going to deplete methionine. So the idea is, what can we add to the diet which may speed up the clearance of methionine? Methionine is very interesting. It's an essential amino acid, and it's the amino acid that's essential for methylating things. The methylation, the adding of a one-carbon methyl group to compounds, is a very important process that happens in many, many areas of bodily biochemistry, including detoxification of drugs that I just mentioned, but also making neurotransmitters, hormones. You can't make adrenaline without methionine. You can't make certain neurotransmitters in the brain without methionine. Methylation of DNA is very important, because when you methylate a DNA base, it puts literally like a stop message on the transcription process. When the DNA message is being read into RNA, and the enzymes are combing over those double helical strands of bases, and they run into this little methyl group that's sticking out, it'll induce it to skip. Oh, don't bother with this gene, go to the next gene. So methylation of DNA bases is important. Many processes, biochemical processes in the body, involve methylation, and so you got to have methylation, so you got to have methionine, and that's essential. One of the things about it being essential, however, is that it's so essential that your body has lots of ways to recycle it and regenerate it. So you don't need much. You only need about 300 to 500 milligrams of methionine every day. That's more like a vitamin than a bulk nutrient, right? But you need it, and if you get just barely enough, you may live longer, right? So that was the aim, to see if we could do something like that. Well, in reading the literature at the time, talking about the 1990s and early 2000s, I guess, is when I got involved in it around 2006, 2007. Looking in the literature, there were other people had discovered really that methionine is a very interesting compound because it's so essential. Every biochemist learned in biochemistry school, right, in graduate school and medical school, we all learned about the, quote, methionine cycle, which is the main way that methionine is recycled when it gets used up. When that methyl group is used up, you can recycle the remaining homocysteine. You remethylate homocysteine, and you've got your methionine back. So the methionine cycle is very key, and every biochemistry book has it. Every biochemistry student had to learn it. Methionine cycle, and this is how you recycle methionine. What I realized was, from work that people had done, but they didn't quite put it together this way, is that when you eat a lot of methionine, as we do, because we have diets that are very rich in meat, fish, and poultry and dairy these days, when you eat a lot of that, your liver is actually spending the next hour or two after you've absorbed that high methionine meal getting rid of the excess methionine. It rather treats methionine kind of like sugar. You need to have sugar, so your body has, if you don't have enough sugar in the diet taken in, your body makes more sugar. It's not essential because your body can make it from other things. But when you have a lot of sugar intake, insulin starts, and the insulin stimulates the body, the liver, and other organs, including muscles especially, to turn on the pathways that use up sugar or that store sugar to make glycogen, or to burn it up in a process called glycolysis. That's what happens when you take in sugar. When you don't have enough sugar, the different cycles happen to make gluconeogenesis, to make glucose from amino acids, to make sugar, so you always have enough. So it's this bimodal kind of attitude that the body has toward these particular nutrients. So in the case of methionine, that was not really recognized, and everyone was always focused on how does the body recycle it, regenerate it, make sure there's enough, make sure there's enough, make sure there's enough. And nobody was really thinking about, wait a minute, maybe we have too much. What does the body do then? Well, it turns out that the most abundant enzyme in human liver, at least these days, seems to be an enzyme called glycine N-methyltransferase. The methyltransferase enzymes are the enzymes which transfer the methyl group from SAMe to some other acceptor, whether it's a neurotransmitter precursor or hormone precursor, a drug that needs to be cleared, gotten out of the liver and all of that. But glycine N-methyltransferase is so abundant. Why? Because it seems that glycine N-methyltransferase is the main clearance enzyme for methionine. How it gets rid of those extra methyl groups when there are just too many and the body doesn't need it. So in that case, what happens when you have a lot of methionine, that methionine cycle that we all learned about, that I talked about, that goes on and on to make sure there's enough methionine, it stops. It goes around, goes in reverse, and other enzymes, kick in. You can see that the methionine cycle has very, very key breakpoints. Certain methionine will stimulate the clearance of methionine if you have a lot around. If you don't have a lot around, it will stimulate the synthesis of methionine or the remethylation of homocysteine to make sure you have enough methionine around. You can't synthesize methionine from scratch in the human body. That's why we know it's essential. So there are actually two things going on. So when I started researching this and writing about it and publishing about it, I didn't have my pathways, just like everybody else has the pathways, showing the methionine cycle in little arrows and small print about this, that, and the other thing. I made two pathway diagrams. This is how it looks when you have a lot of methionine around, and that is how it looks when you don't. And you can see the key breakpoints, switches. This switch turns on, that switch turns off, and it goes this way. That switch turns on, this switch turns off, and then it goes that way. It clears methionine or it resynthesizes methionine, like that. So that's how I deal with the biochemical cycles. Remember, again, that those other pathways you were talking about, the mTOR, and those are signaling pathways. I'm not talking about signaling pathways. I'm talking about the sort of bulk nutritional intermediate pathways. How to generate, what your body needs in terms of fuel, fuel for methylation as opposed to fuel for just motion in general. That was my focus. Now the other focus in terms of why methionine restriction works to promote, prolong lifespan, we still don't really know. We know part of it because I tried it. I thought, well, okay, glycine. So here's the basic simple hypothesis. Let's try adding glycine to the diet, because if glycine seems to be the receptor of methyl groups for the clearance pathway, maybe we can help clear it and mimic the effect of methionine restriction and make the animals live longer if we just add glycine to the diet. So what did we find? We found it made the animals live longer. Mm-hmm. Yay, we've mimicked methionine restriction. No, we didn't. It didn't make the animals live that much longer. It was maybe 10%, 5, 10, 15%, not 30 to 50% longer. Okay, that's one thing. The other thing was that it made the animals live longer, but it didn't change the amount of methionine in their liver or in their blood. Well, if it's not going to change the amount of methionine there, then it's not increasing the clearance of methionine. That's going on independently of how much glycine you have in the system. So just adding more glycine doesn't do what we were adding it to do. So that's where you reach a fork in the road when you're an experimental scientist, right? You say, oh, okay, well, hey, it looked like it made the animals live longer, and you look a little further and you see the concentration of methionine doesn't change, and say, but it didn't work by the same, whatever the mechanism is for methionine restriction. Oh well, oh shucks, throw it out, let's start from scratch. Or you say, but this stuff it made the animals live longer. There's something this does that's beneficial. What's going on here? Right? So that's where I got on the glycine bandwagon, or started driving the glycine bandwagon, back in about 2008, I would say.
Wow.
So we're nigh onto 20 years now, since I started working on it, maybe 2007.
Quick question though, before we get into, because you are driving that bandwagon, but why do you think, is that the reason no one else has because even in this paper is a very brief summary about glycine, and you said there was even maybe some inaccuracies about glycine. So all the other scientists saw this and said, headed the other way, I'm still going to look at methionine. But what made it different?
Oh, I don't know that they saw it and headed it the other way. I think they I don't know whether anyone noticed. So I couldn't say that.
Okay, maybe just not exciting enough to.
Yeah, well, yeah, it was just. I mean, once you have this result, it's not like once we got the result in our laboratory, the world knew about it instantaneously, right? I mean, it took a while, and I think I published the abstract in 2011, actually, where it says in the abstract, that the aim of it was to add glycine to act as an accelerant, if you will, to accelerate the clearance of methionine so that it could produce the effects of methionine restriction. When you submit an abstract for publication, they accept it. That's in the abstract. That's what gets published in the book of abstracts. And then when the meeting comes around where the data are presented as a poster or as a talk, you've done more research on it by that point. It's some months down the road. So by the time I got to presenting the actual paper, the actual abstract as a poster, I guess, at the time, we had to admit, no, it doesn't work this way. So it was kind of obscure. I mean, it was available for the world to see it, but I think it was just kind of not really, noticed. But I'm saying, as a phenomenon generally, when scientists are in their own laboratory looking at something, and you find a result that's an interesting result but not interesting enough because it's sort of off the beaten path of what you were looking for, then you just ignore it and you move on. So I think that's what's the case with lambing. Lambing is still looking for what is the driver, what is the reason for methionine restrictions increasing lifespan. As I say, part of the reason is because it takes glycine. As I say, when you clear excess methionine, you use up glycine. So if glycine is a real important compound that you need to make sure you have enough of, and you have too much methionine, and you're using up that glycine to clear the excess methionine, that's probably one of the reasons that depleting methionine helps, because it stops the depletion of glycine. So that's one of the reasons it works. But the main intrinsic mechanism, whatever it's doing in all of those signaling pathways like mTOR and and all of those signaling pathways, that's still not really known, which of course Lambing admits. We don't really know what that is, and he's still working on it, and lots of people are working on it, and that's still fine. But I'm not working on it because I found out how important glycine is. And so I'm working on the glycine angle. It's a different angle. So I basically segued. I switched on to a different track because I found a new track which was very productive, even though it was not productive in terms of answering my original question.
Nice.
So that's where I would say the science, scientists, I think it helps to be a little more flexible because you find out more things. Just like writing the pathways, you scratch your head and your eyes go bleary and you can't quite figure out because all these intricate pathways are so confusing. Instead of saying, wait a minute, let's draw up the pathways and see what they look like when you have lots of methionine around, and let's draw up the pathways and see what they're doing when there isn't, and see the differences, and then it becomes really, really clear.
And I love that you were open-minded enough to say, oh, I got to go over here. And I do think the future is, right, one of the big mistakes here is looking at protein as protein and not looking at amino acids as very individual, not only building blocks, but signaling molecules. And originally when I saw this paper, I was like, oh my gosh, methionine restriction, like you're saying, at least partially, is probably preserving glycine.
Yes, partially that. Yes, that's clear. I mean that's kind of a no-brainer at this point. But most of its effect, the majority of its effect, which has to do somewhere along the line with signaling that says we don't have enough of this essential amino acid to grow. Yeah. So we're going to conserve that and stop growing and just function. Because let's say all these creatures that God put on God's green earth, our function is not necessarily to extend our own lifespan.
Yeah, right?
Especially because there is evidence also that if there is a reproductive disorder, like in some certain cases of dwarfism, where the pituitary gland does not develop fully or properly such that the reproductive system is not matured to start functioning as a reproductive system, you actually get the animal lives longer, and actually people live longer. Dwarfs who have that kind of dwarfism, where the whole reproductive system, the generation of female cycles, or of the male spermatogenesis and the female oogenesis, and all of those cycles that generate the ability to produce offspring. If you shut those things down, you can actually extend lifespan. But that's kind of not the way God intended it, because we're being fruitful and multiplying is one of the, it's one thing to be fruitful and multiply. It's not be fruitful and multiply, and by the way, at the expense of living as long as the individual possibly can. I mean, that's not what's built into, animals or animal body. We have animal bodies, our bodies, or the bodies of the animals that we use as models in the laboratory. So it's another point of view, the perspective of, well, of course, a body that functions. A lot of people will go back to the natural function of the body. You think, oh, the natural function of the body, the way God intended it and everything, is to live as long as possible. Not really. It's to be fruitful and multiply. And that you sacrifice, as every parent knows, you sacrifice some of your individual substance to raise your children. In the same way, the body loses some of its potential for individual longevity by reproducing. So, for my own very limited knowledge of the signaling subject involved in all of this, I would defer to someone like Lamming about it. I would say, the body is not necessarily designed to live as long as possible. So when you're doing that, it's not necessarily what do we want to make more. It's not necessarily more natural to live forever, although that is a fundamental aspect of life, is that everything that lives wants to go on living. That's part of the spark of life that we can see in anything we recognize as being alive, that it wants to keep on living. But that's not necessarily what's built into the system of living on Earth, because you have to reproduce. Otherwise, the whole species is gone. So it's somewhere along there, and I would, in deference to these guys, I think, they're humble enough to admit we're not really there yet. We don't really know what it is. And once we find out what it is, we won't necessarily want to pursue it. As I said, a methionine-free diet or a low methionine diet is very, very difficult to stay on, and it doesn't necessarily make life worth living.
No, and what if it was like a methionine and glycine balanced diet? Not that in equal proportions, but like we eat enough glycine to balance the amount of methionine.
Here's the trick. Yes, and you will still get the benefit of whatever the lack of glycine clearance benefit is from methionine restriction. But you still will not get the benefit in terms of lifespan extension of methionine restriction. To do that, you have to be you can't just, it's not like, okay, we normally eat, let's say, 2 grams a day of methionine. We only need half a gram. So if we cut it down from 2 grams a day and we make it 1 gram a day, or 3/4 of a gram, or 1 1/4 grams, that should make, it doesn't really matter. You really, really, really have to get down to a very restricted intake, a really artificially low intake of methionine before you see this effect. And as I said, it's not really natural. Right. It's not natural for rats and mice to live an extra 4 years and not grow to a bigger size, right? As I said, they don't grow to normal size. That's a key thing that people need to recognize, is that, you say, well, now that I'm full grown, wouldn't it be nice to restrict my, growth and have a methionine restricted diet? You can do it, but it's a very, very difficult thing to do. So anyway, I decided to just get on the glycine wagon, as I said, or to start driving that bus. And, it's what I found out was, that glycine is the most extraordinary compound that has not been fully investigated. That's been ignored because it's viewed as non-essential. But I view glycine as really the bridge between the non-living and the living world. Glycine is made in the non-living world. You can find it on comets and asteroids long before anybody walked the Earth or any other planet, in case there's life on there. It's made inorganically from carbon dioxide and ammonia and formaldehyde, very simple building blocks. Your own liver can make it from those building blocks. And yet, you have to say it's an organic compound. It's an amino acid, right? It's, amino acid proteins are only made by living things. You don't find proteins in the non-living world, but you do find that amino acid and all the other non-essential amino acids. Or not all the other ones, but you do find glycine. That's the only protein-making amino acid that you find in the non-living world is glycine.
Oh, that's very interesting.
I mean, taurine is another one, which is an amino acid that's not used in protein synthesis. But of all of the 20 amino acids that are used to make protein, glycine is the only one that you don't find in the non-living world, and that's why glycine is glycine is glycine. There's no, lots of people sell L-glycine, which is,
Yeah.
it's a joke, really. They don't intend it. They think they're selling the good stuff. Oh, it's L-glycine, But L-glycine, there is no L-glycine. Glycine is glycine. There's no left-handed and right-handed glycine because it is made inorganically. That refers to the function or the existence of a particular molecule. If the molecule is so simple and symmetrical that it is identical to its mirror image, then there's no L and R, no left and right. Right, you have a left and right hand. Why? They're not mirror images of each other, are they? I mean, they are in a sense mirror images. They're different, right? You can't wear a left-handed glove and a right-handed. I mean, they make them general enough so they sort of fit, but they're different. So the mirror image of your right hand is like your left hand. It's not a copy of your right hand. So it's the same thing with any kind of molecule. If its mirror image is not the same as itself, then it has chirality, which is to say handedness, left and right hand. Left-handed glycine, right-handed glycine, but there is no difference. Glycine is glycine. So beware of anyone who's selling you L-glycine. It's the good stuff, charging you more for it. You might wonder if they're actually selling you glycine if they do that.
It's a good question. Can you tell us though, what is your major discovery around glycine?
The major discovery, well, that's a whole other, that's where it led to me was, because we understand now that most diseases that make us sick and die these days are rooted in chronic inflammation, and nobody knows why. There's another big article came out of the New York Times Magazine that we can talk about later, if you want. I think that the Times article said basically that it will be when we discover it, when we discover why inflammation becomes chronic and people suffer this chronic low-level inflammation, it's so important health-wise that when that's discovered, that will be the health discovery of the century.
Yeah.
Well, maybe it is the health discovery of the century, except I already discovered it in 2010. So it's still this century, I guess, but they don't know it yet. They're at least 16 years behind the times. But so what is that discovery? That discovery is about inflammation. Inflammation is believed to be a natural response to injury. Yeah. Infection or injury. Then any book you see, except mine, that defines inflammation will tell you that it is the immune system's first response to infection or injury. And I found out that is not true, categorically not true. First response to infection, yes. To injury, no. It does no good in injury because all it's doing is secreting poisons to try and kill something that isn't there. And in doing so, it just does damage to normal tissue, which causes pain and swelling and cardiovascular disease and brain dysfunction, and all these other degenerative diseases, which are from low-level chronic inflammation. Why does it happen? And I found out that it happens because of glycine deficiency, because of a key role for glycine, not as a protein-making amino acid, but as a key intermediate small compound which acts in a micromechanical way to stop the cells that cause inflammation from doing so when there is no living microbe around that needs to be destroyed. And so these cells that do inflammation when there's bacteria around or viruses around or something like that, they get activated, and they get activated really only because there's not enough glycine around. And why is this something new? Why are all these diseases, cancer, heart disease, all the rest of it, why are they? They've always been with us because the body really does need to take in a certain amount of glycine. It can't make really enough. But now modern living and modern dietary habits have changed things so that we're now really, really glycine deficient. And now there's not enough glycine bathing these immune system cells to make them behave and not cause inflammation when there's no infection, when there's only injury. So that's really the main discovery is about inflammation, what its actual function is. And it's all about the glycine, because glycine it's like a trigger lock on these cells that cause inflammation. Inflammation is caused by the first responders, and the first responders have dual roles. A lot of researchers understand this now. A lot of times there's cells called macrophages, and they understand now that macrophages are not just in the blood or in the lungs or in certain tissues. They're everywhere. Macrophages are like an army reserve, a reserve security force that you find in every organ with certain specific characteristics that make it suitable for a particular organ. So there are liver macrophages called Kupffer cells. There are lung macrophages called alveolar macrophages. There are brain macrophages called microglia. There are macrophages in muscles. There are macrophages everywhere you look. You find macrophages, which are cells of the immune system, even though they live with and they look like cells of the organ that you find them in. It's kind of like the way I teach this or taught this to my students before I retired is very simple. I say, well, take our college. It was the one original building of the college, which is where I taught it. So here's the building of the college. I said, let's say the college is the body, and everybody who works in it, right, who spends time in it, is a cell of the body. So what kinds of cells are they? Well, most of us are student cells, right? Most of the people in the building are students. Okay, but you don't just have students. What else do you have? Well, you have faculty, like me, right? You have faculty. And, well, it's not just faculty, but you also have what? You also have administrators, right? Guidance counselors and, various kinds of other administrators and secretaries and so on, which process all the grades and the courses and the curricula and whatever. All of those are different types of what? Educational professionals, right? We all have degrees that are in education itself or in this aspect of education, in science, in math, and whatever else we're doing in the humanities. So the cells, quote, of this body that is the building, that is the school, the college that we're in, is filled with lots of cells. Most of them are students, and they're also faculty and administrators. All of us are education professionals of some kind. You can say a student's not a professional, it's in training, but we're all in the education business. Well, is that it? There's nobody else who works in this building? Oh, yes. What else is there? Security. There's security guards, the people who check your ID when you walk in. There's the maintenance guys who change the light bulbs when they go out, right? And who fix the plumbing when it goes bad and everything like that. So there are other cells, people, which are involved in maintenance and repair and security. That's what the immune system's macrophages are. They are there for security and maintenance and repair. So they are like the first responders. Now, if you go out there in the larger world, and let's say, my favorite analogy is where you go out on the highway and let's say there's a pileup on the highway, an accident. Right, traffic is all snarled up. So what happens? So who comes? So the police come, right? You hear the sirens, the police come. They call for other first responders, right? Could be the fire department with ambulances and ambulances, fire department, police, right? First responders. So what do the first responders do? Well, they divert traffic. They take accident reports. They call up the ambulances to take away the injured, and all of them take and body shops to take away the injured vehicles, all of that. But all the while, all those state troopers that come around show up at the accident, they got guns on their holsters. They're armed. But do they come out and draw their weapons and start shooting up the place? Well, of course not. Not unless there are bad actors present. If there are criminals around or terrorists around or something like that. Then they may draw their guns and need to shoot them. So their first responders have these two different modes. I call them the rend mode, to destroy things, break things up, shoot things, and the mend mode, to fix things. The same cells do that. The macrophages, they can be in rend mode or mend mode. And the official designation for these actually in cell biologist and immunologist lexicon is M1. M1 macrophages are armed and dangerous, right? They'll secrete poisons. They will actually do inflammations. They will precipitate the inflammatory cascade, which shoots the place up and causes cellular damage so it can kill the infection, if it is an infection, before it kills you. But then there's this M2 mode, the mend mode, more like the ambulances. But it's also, but the cops will also call them too and never end up drawing their guns if it's just an accident. So that's what you have with macrophages. Inflammation only happens when the macrophages are M1. A lot of people, clinical researchers and so on, will tend to still think of wherever you see a lot of macrophages around, that they call that inflammation. Even though it's not really inflammation, it may just be an infiltration of macrophages, maybe around just to fix things and not break things. So the same cells will fix things or break things, depending on what mode they are in. And what's the difference in what sets it up? Well, there are particular receptors that will tell these cells, oh, there's bacteria around, you better draw your weapons, right? So they typically experimentally will use substances called lipopolysaccharides, which are substances that you find in the cell walls of bacteria that identify, oh, bacteria around, draw your weapons, right? So they'll use experimentally, they will inject lipopolysaccharides, not live bacteria, but lipopolysaccharides to elicit this M1 response and actually cause the inflammatory cascade. That's what will elicit it. And then, but if you don't, they will stay in they would just be activated to come to the scene and clean the place up, to gobble up dead cells and cell debris and so on, and help to initiate the healing response, secreting certain factors to attract new blood vessels and stimulate growth and that sort of thing. So there are these two different roles that the macrophages play, and the thing is, what switches them from one mode to another. It has to do with electrochemical switching at the surface. There's actually a voltage at the surface of the cell. On average it's around 70 millivolts, not 120 volts like you find in a wall switch to turn a light on. But it's the same principle. If you have a light switch on the wall and the light is off, you have 120 volts on one pole of the switch and 0 on the other, right? The switch is open, it's disconnected, it's in the off position. When you put the switch in the on position, now you close the switch. Now both poles of the switch are connected, the voltage between them is 0, the current flows to the light goes on. So this is basically what happens at the cell surface. The cell surface is polarized. It has this small voltage. Now, while the cells are going around doing everything, phagocytizing we call it, gobbling up dead cells and cell debris, there's a lot of cytoplasm moving around and membrane moving around, and there's a lot of leakage of ions. And it's those ions which are different on the inside and the outside which maintain this voltage. And one of the mechanisms for allowing the voltage to stay high is to allow the influx of chloride ions, because normally the inside, in the resting state, the inside is negative with respect to the outside. But if you open up these special channels that allow the negatively charged chloride ions to flow in, it will keep the negative charge elevated on the inside so that it doesn't get that signal to start, okay, we're M1 now, let's draw our weapons and start shooting. So those channels are actually glycine-gated chloride channels. And the glycine-gated chloride channels, they were identified many decades ago in the central nervous system as part of neurotransmitters. But only in the 1990s, that's only a few decades ago,
Yeah.
were they recognized by Ron Thurman's group in UNC, University of North Carolina. He passed away, unfortunately, in 2001. But a lot of his work was very, very important in determining that this is what activated macrophages. They used lipopolysaccharide and showed the activation or the lack of activation, and shows that glycine is really acting like a trigger lock to prevent the activation to the phagocytic, to the inflammatory state. This is really the main discovery, my main contribution to the field of human health in general. And that's really was the bigger target. I was working on a particular thing, but when you notice something else, say, wait a minute, you don't want to get distracted by something that's not important. But don't assume that it's not important when it's when you see, take notice. See, a lot of people that say, well, Brind stumbled across this. No, I didn't stumble across anything. Because as I like to say, you stumble when you're not paying attention. You discover things when you are paying attention. What they mean really is that you weren't expecting it. When you run an experiment, you say, okay, here's what I expect to find. Did I find A? Yes? Yeah, no. It's not quite A. Didn't work. Experiment didn't work. Throw it out. Let's start again. No, but I found something else. I ran the experiment and it showed me something happened, but something I did not expect. And that's why they say stumbled, but it's a surprise. In other words, it's a surprising finding. don't ignore it if it's an important finding. See if it is or not. It's worthy of your investigation. So that's what I did, and ended up finding out this is really the key to inflammation. And I decided I discovered that really in 2010, and by the end of 2010, that's when I started this business, because I realized that I could spend the rest of my life and more with other people taking my place to try and convince the scientific academic world that what I has found was real, and that it means what I think it means, and then you prove it this way, and prove it that way, and prove it any other way. But if I know that it works, and if I know that it is effective, and I know that it's safe, and the FDA says these things are safe and harmless and non-essential and all that, well, then I can sell it. Because if I can sell it, make a product, so I made the product, Sweet Amino, and if I can make this product and people buy it, now people can actually take advantage of this discovery. What is the point of someone curing cancer in 2010 if nobody finds out about it until 2050? So, now, I'm not saying this doesn't cure cancer. It doesn't really, its effects on cancer are it probably slows the development of cancer. It slows the growth of certain cancers, but in general, it's not really directly related to cancer.
I would imagine, and Sweet Amino is one way. We all have a glycine deficiency, but do you think then, would it be fair to say that this glycine deficiency and the escalating rates of unnecessary inflammation could be more important than trying to restrict your protein and your methionine in terms of living a healthy life? What do you think? It seems like the better way to go.
Yeah, let's say, would it be better for the average person in society to live to 100 and be more healthy but miserable, or to be merely 85 or 90 and have a full life with a varied diet and and all of that?
The other thing that bothers me is, in order to restrict methionine in animal products, you have trouble building muscle as you get older, and the quality of your life goes down when you can't build muscle and you can't be active. So it seems like if you could keep your protein in the diet, but also get adequate glycine, then you could maintain a quality of life where less inflammation, more muscle, more mobility.
And again, I say what we found out, remember, that glycine does not interfere with your ability to utilize methionine or any other key amino acid. It doesn't, as we originally thought, it does not serve to accelerate the clearance of any amino acid. It just has its own role to play, its own several roles to play, really, which you need more of it. And because we now, remember we discussed this triple whammy before, that, what's happening in the, the 21st century here in the modern era, what's happening? Well, we're eating more and more and more methionine because we tend to eat more. So what that does is it depletes glycine. Well, so that's not good. But we're also eating less glycine because we're throwing away the bones and the connective tissue. Strike two. Strike three, we're eating so much that we're getting fat, and when you get fat, what do? It turns out, we know now from recent research, that when you get fat, it actually severely inhibits the synthesis of glycine. So that obesity is one of the conditions of more severe glycine deficiency. Yeah. So we eat more muscle meat, we eat too much overall, so we eat more methionine, we eat less glycine, and we eat so much we get overweight, so we make less glycine. Strike three.
Yeah.
Where a door is open now for all manner of chronic disease.
Well, yeah, and it's an amazing finding that you have, and we are trying to help people understand just adding glycine and potentially reducing this unnecessary inflammation. It's amazing. Now, I just want to close this episode with a few just quick questions. So I think this protein restriction stuff's interesting, right? There's something there, maybe linked to longevity. Are you going to ever restrict protein? Do you think there's a case for that at any stage of life? Because I know you don't want to restrict protein when you're pregnant, obviously, when you're developing, when you're recovering from injury, if you're trying to build muscle, if you're an athlete necessarily. But if there's people who are older, do you think there's ever a case for someone restricting protein? Are you going to do it?
Well, there might be. I mean, and I as a scientist, I like to think as a good scientist, I'm not going to rule anything out.
Yeah, I love that about you.
In terms of restricting protein, my objection to it, again, is protein in general. It's
Yes.
got to be, something a little more specific than that. Although there are conditions where, disease conditions where you need to restrict protein, where you need to restrict fat. Why? Suppose your digestive system is missing some enzymes that you need to digest protein. Suppose you have a pancreatic. I see ads on TV now for some drugs aimed at people with pancreatic insufficiencies, where they don't make enough enzymes to digest certain foods. So proteins, where one protein is pretty much just like another protein, one of the things that all the proteins share in common is how they are digested by which organs in your digestive system. So there may very well be cases where you need to have a lower protein diet. In some cases, though, you may have to take hydrolyzed proteins, so you may have to take just the amino acids. You can take the amino acid mixture, right? Mm-hmm. And not have proteins. People who have had stomach cancer, who have had their stomachs removed. Oh my goodness, a vital organ. No, stomach's not a vital organ. It's a digestive organ. So if you give the digested food that would otherwise ordinarily be released after digestion from the stomach, if you give basically the products of stomach digestion, you don't need a stomach. And as technology has advanced and the knowledge of what's in foods as nutrients has advanced, more and more things that used to be essential organs are less vital. So essential organs, we call them vital organs, organs without the function of which you would die. So a couple of 100 years ago, what was another vital organ? Teeth.
Yeah.
When people they got old and decayed and they lost their teeth, they went off to die. They couldn't eat anything because, Mm-hmm. So unless you have enough means, we didn't have magic bullets, or blenders. You can just, take we go out hunting and gathering, and we take whatever we have, we put it in the blender, grind it up, and sip it through a straw. We didn't have that 500 years ago.
Right?
So at one time, teeth were essential organs. And then stomach was, so the more we can reproduce the effects of particular organs, it's like, and what happens if you have an artificial heart? When there's an artificial heart that works as well to pump blood as a human heart, what does that mean? It means your heart now, that's not an essential organ, right? Because you can replace it. So I would say, yes, of course there are cases where the restriction of certain kinds of nutrients, be they proteins or whatever, may be indicated for different disease conditions. But again, to say protein in general in terms of the nutrient molecules we are obtaining, too much of, too little of, wrong balance of, right? In terms of those things, you can't just say protein or not protein, more protein or less protein. You have to say, well, what kind of protein? Is it plant protein or animal protein? Is it animal protein? Well, you mean all the animal? You mean all the glycine that's in the bones and connective tissues? Well, usually not when they're studying these things, in scientific studies.
Yeah, and I want to clear this up before I go to my last question, which I'm really excited about. But people got confused last time. You're not saying plant proteins are better or that veganism is better, only that when you're eating animal proteins, you should be eating the whole animal, right?
Or, yes, that's right. Yeah, the nose to tail, or don't eat the animals at all. That's true. That's fine. You don't need to eat animals. Vegans can live fine and happy and healthy and long. And in fact, many studies going back decades and decades show that vegans tend to live longer. They tend to have later onset or lower incidence of cancer and cardiovascular disease and so on. And my hypothesis, which I think there's a lot of evidence for, is that basically that's because when you compare plant protein to animal protein, you're not including the bones and connective tissues, which have most of the glycine. So when we eat an incomplete animal protein, things do tend to get out of balance, and that's why meat, fish, and poultry eaters do have more and earlier onset of these chronic diseases because they have less glycine in their system. That was proven in 2015, right? By that
in the EPIC study.
EPIC study, yeah. They didn't even know what they'd proven, but they proved it nonetheless.
I love that. And my last question for you, before you tell everyone where we can get sweet amino, is this. I always love that the science is evolving, right? We're not saying this is the answer. We're saying, hey, look at the science, it's evolving. I'm kind of interested in, I was listening to this interview with this woman named Tracy Anthony. She's on the Dr. Gabrielle Lyon podcast. And Dr. Gabrielle Lyon, she came on my podcast. She thinks cyclic protein restriction is the future. Now, again, what proteins? So what she was suggesting, what they talked about, which I thought was really interesting, was potentially bringing in something like a glycine-rich protein like bone broth, So reducing the methionine, reducing the branch chain amino acids, and maybe like 1 day a week, 2 days a week, having a low methionine and leucine and isoleucine and valine type diet that floods your body with, you're still getting protein, but you're getting that glycine rich protein. So kind of like a bone broth fast day, once or twice a week.
Right, that's the word, I think. This is really approximating a variant of intermittent fasting, isn't it? And intermittent fasting is good for you, yeah.
Exactly. It's bringing that, if there is a benefit to that protein restriction for proteostasis and allowing those proteins to kind of be rebuilt, or the broken ones broken down, or, there's various
Yeah, it's a way of doing intermittent fasting without really fasting. Just a partial fast, like like Lent is, right, in a religious tradition, right? Lent is like that. It's an intermittent fast of certain kinds of foods, and all of these things they're good for you, apparently spiritually as well as metabolically. I mean, it sounds like a reasonable hypothesis. Of course, the proof is in the pudding. How do
But what I
things
love
work?
about it is that it not only does that, gives us that protein restriction, but also that glycine adequacy. So I don't know, like, is it a way to like rebalance that and make sure that you're having enough glycine and also potentially benefiting from protein restriction, but also not necessarily not building muscle. So on the days when you have hard workouts, right, where you're recovering from some sort of injury, you keep that protein high, even the methionine rich. But then on days when you're more sedentary, perhaps, you can bring those amino acids down, do a bone broth fast, maybe once or twice a week, and it is a reasonable hypothesis, yes?
Yeah, I think it is, and we should also keep something else in mind that tends to be forgotten. I remember there was a paper that I read some years ago in the Science News about, they were looking at some vegetable that was grown on the prairie as potentially, because it had high protein in it, as potentially a replacement for meat, fish, and poultry. This school of thought that we can, not just eat worms and what, but eat other things, that there are other plants out there that may be able to substitute for that kind of nutrition. And it was a whole long paper about it. Has a content of this and the content of amino acids and the contents of some other, plant flavonoids and isoflavonoids and whatever else it had and all this kind of stuff. And there it was, the very end of the paper, I got to it and I just stood there and as if I could talk to the authors, which of course I couldn't, and just thinking, I had this question like, what does this stuff taste like?
Yeah.
I mean, and the reason, in a biochemical sort of way, is if you wanted to get the most potentially sophisticated, accurate, long-lived, robust nutritional sensor that you could possibly build or design, what would it be? Well, how about the human tongue? That's very sophisticated. So, these days, the problem with processed foods is so much sugar and salt and other artificial flavors are added to things that they are in many cases deliberately intended to confound our natural nutritional sensation, right? So your tongue tastes sweet. Oh, that's sweet, so it's got sugar in it. Okay. Well, now you're eating too much of it. So what do? Oh, well, we'll just put in something that tastes like sugar, but it doesn't have all the calories and it won't stimulate blood sugar rise and diabetes and all of that. So you have the whole artificial sweetener injury, whose job is to confound that wonderful sophisticated biosensor and make it not work right to detect what. So one of the questions I like to ask my students is when we talk about the digestive system, the first thing that happens in the digestive system is that you have salivary amylase. In your saliva, it actually begins working on the food right away, at least carbohydrates, and it will digest carbohydrates and turn starch into sugar. So salivary amylase is an easy enzyme to work with. We can use our own and do experiments with it, and that's great when you're doing, introductory human biology. Okay. So now we have this salivary amylase, but here's the conundrum. You read the book, and it talks about salivary amylase works at neutral to slightly alkaline pH. As soon as you get into the stomach, the pH is strongly acid. So the amylase, so that you're chewing up this, a piece of bread, right? You take a bite of a roll of bread and you chew it up and you swallow it, and all the while in your mouth and all the way down your esophagus on the way to your stomach, which only takes a few seconds, of course, it's the salivary amylase is breaking up starch and turning it into sugar. And then what happens? As soon as it hits the stomach, the enzyme is degraded. The enzyme is denatured, doesn't work anymore. That digestion stops. And it doesn't start again, it's very incomplete at this point. It doesn't start again until that nutritional mix, that bolus of, stuff that you just ate gets into your small intestine, because then your pancreas secretes into the small intestine pancreatic amylase. It's basically the same enzyme as salivary amylase. And that continues the process. And so people ask the question, why did God make the digestive system such that you actually have salivary amylase when it's such a waste? It's so inefficient, because that enzyme just gets destroyed as soon as it hits the stomach, and now the pancreas has to make amylase and start all over again digesting the starch, finishing the job. So the answer came to me sometime so many years ago that I'm always teaching my students is that you have to remember that inside all these clothes and contraptions that we have and dress ourselves in, we're all cavemen and cavewomen on the inside. So think about being a caveman and cavewoman, and now, first of all, you get up in the day and you don't have a hearty breakfast, because who has a hearty breakfast? You're a caveman. You just got up. You got no food. First thing you have to do is go out and hunter-gather, right? You got to go find something to eat. In fact, that's why, they say breakfast is the most important meal of the day. Baloney. That was a fabrication by the Armour Meatpacking Company. Armour Meat Company in, I think, 1927 or something like that, with an advertising campaign which said, breakfast is the most important meal of the day, so you would... And so they wanted to sell Cheerios or whatever else they wanted to sell bacon and sausage. So breakfast is the most important meal of the day. That was their advertising campaign, and that stuck. And now we all have a hearty breakfast. Even I have a hearty breakfast, which we've adapted our situation to do. But let's say you stopped working and stopped doing everything when the sun goes down, maybe 5, 6, 7 o'clock, depending on the time of year and where you live. Right? Now you're done with your hunting gathering for the day because it's dark outside, so you go to sleep. Sun goes down, you go down. You wake up in the morning bright, and now you haven't eaten for 12 or 14 or 15 hours, right? And what are you supposed to do? You got to have breakfast. Well, about an hour before you get up, this is endocrinology now, about an hour before you get up, your brain secretes a hormone called CRH, corticosteroid-releasing hormone. Right? So the hypothalamus in your brain secretes corticosteroid-releasing hormone, which stimulates your pituitary gland. And your pituitary gland now, in response to that, secretes ACTH, adrenocorticotropic hormone, which is basically cortical-stimulating hormone, which stimulates your adrenal gland to make cortisol. And cortisol tells your liver to make you breakfast. And it tells all the other organs, because it's a hormone, circulates throughout the whole body, it tells all your other organs to supply the amino acids, the proteins, as the raw material to make sugar, so they can go back to the liver. Your liver makes the sugar. Now your liver makes you breakfast. So because of the cortisol spike, from the CRH spike to the ACTH spike to the cortisol spike in the morning about an hour before you normally get up, and it will change. If you work on the night shift, after a few weeks, your cycles will change. Your liver will then make you breakfast, and that's the natural way of things, to have this intermittent fast. So the best intermittent fast is after dinner, don't eat anything else. Don't have breakfast, get up, go to the gym, do whatever you do, have water, stay hydrated and all that, and then eat when you're done, eat at like midday. Now you have a nice long intermittent fast every day. Now, I don't pretend to be able to stick to this regimen. There was a while that I did more or less, and I still more or less have, you have an overnight fast, and that overnight fast is very good. That's intermittent fasting. So now intermittent fasting is a whole new thing, right? It's like somebody just invented intermittent fasting. No, your body's actually made that way. That's what it's designed to do. And so when you discover that intermittent fasting is good for health, you're really discovering or rediscovering one of those habitual arrangement of activities, organization of your activities in terms of eating and exercise and sleeping and all of that, that is in tune with the way your body's actually designed. So when you're a caveman or a cavewoman, remember about the amylase story, which I never quite finished. When you're a caveman going around hunting and gathering, and you're looking at plants and you're pulling up roots, and maybe you pull up a carrot, and say, oh, that's an interesting plant. Oh, it's got an interesting orange root. You give it a taste. Not bad. It has a little sweetness to it. because you chew it a while and the starch in it breaks down. And then let's say you try arrowroot or something like that, which has a non-digestible starch. Because a lot of plant starches are not digestible. You taste that and it's nothing. Yeah. Your tongue is telling you it's not nutritious because it didn't get sweet. So that's why you have amylase secreted in your saliva. It's a biochemistry laboratory. It's an assay. You're going around being able to assay the nutritional content of your food. And that is the instrument, your tongue. And whatever supplies the chemical reactions needed to make things sensible to the tongue. But we get so involved in our test tubes and our electronics, and we think we invented everything. It's like we're just rediscovering the way God made it. It's really all it is. It's a wonderful journey to discover that, but we tend to lose track of that, or these days outright deny it. Especially because I got a PhD and I have 50 years of experience in the laboratory and I've written 300 peer-reviewed papers, and I'm an expert on this and on that. When you ask the expert, if you're an expert on cancer, for example, if you're an expert, tell us, what causes it, how do we treat it and get rid of it. You're an expert, so you should be able to tell us. If I call up an expert to fix my toilet because it's not flushing right, and the plumber comes over, he's an expert, and I expect him to fix it.
Yeah.
If you call yourself an that's why I don't like to call myself an expert, because if you're an expert in some area of medical research like cancer or heart disease, because certainly heart surgeons, you call up an expert in heart surgery and you say, you need a new mitral valve, you expect them to be able to fix it. Well, okay, they'll fix it. I had a surgeon a few months ago fix a hernia. I needed a hernia repair, and she fixed it. She fixed it. She had to resect it a little and sew it all up and everything like that. She's an expert. She fixed it. But then you have these guys going around, I'm an expert in cancer research. Really? If you're an expert in cancer research, then the problem is solved.
You're right. Yeah.
Then you can fix it.
Got a lot to learn there.
Yes,
yeah.
there are really those things which are still unknowns. We really don't have any experts. We have, people like me we're seekers. We're really students of the human body and all of these systems which exist for our benefit. And when things go wrong, we're trying to figure out and we have a hypothesis and we test the hypothesis and we may accept it or reject it or modify it or move on to something else or find something even more important. I have the hypothesis that when penicillin was discovered, and Alexander Fleming invented penicillin, again they say, he stumbled on it, because he found out that he had bacterial cell cultures and he found these clear spots in the bacterial films, which meant the bacteria were dying. Something was getting in there that was killing bacteria. And found out it was something, he had mold cultures, and some of the mold products were getting into the bacteria and causing the bacteria to die. So that's how he discovered penicillin, that was what was getting into his bacteria. But my hypothesis is that there were probably 100 other scientists around that time or before that time who found the same thing going on and said, crap, my bacterial culture's ruined again, and threw it out in the trash and didn't learn a thing, But this is what happens. So, the human mind, we are all subject to this kind of frailty, but we have to be conscious that we are, when we're investigating, we got to avoid tunnel vision, avoid getting so hyper specialized and so knowledgeable about some particular pathway. This guy's an expert on mTOR. Welcome, I'm an expert on,
Dr. Brind.
On N kappa beta, whatever, and I'm an expert on autophagy, and I'm, everybody's an expert in some tiny little area of cellular function, and the danger is that you really lose sight of the whole endeavor of trying to rediscover what it is that makes us tick and tick longer and tick healthier, right, and all of that.
Well, thank you for being that person who, sees the bigger picture and is willing to press forward even when it's not what you originally planned on studying. So Dr. Brind, you've made a supplement called Sweetamine. You can get that 8 grams a day. You can put it in your tea, in your coffee. We do bone broth protein powder that doesn't taste like, bone broth. There's collagenous cuts. There's just eating the whole animal.
These are all ways of getting glycine, yes. Getting enough glycine because, the typical diet is short about 8 grams a day. And
Yeah.
so, but glycine is sweet, so you can tweak it a little bit and make it taste just like sugar, and it doesn't give you any calories and all that.
Yeah, and so give me your last little line here. Where can people find Sweetamine?
Oh, sweetamine.com. Where else? SWEETAMINE, sweetamine.com. Simple, you can order whatever you like, in terms of quantity, including if you use the, what is the promo code? Is it Autumn? Is that the promo code? Yeah, if you use the promo code Autumn, it's got one of those silent N's in it, right? Autumn.
Autumn.
AUTU. You got to put the N on the end of autumn, or it won't work. You get $5 off your first purchase, whatever you want to buy. So you can, which means you can get a 12-day challenge, 12 packets, daily packets of Sweetamine for as little as $14.95 if you use the promo code Autumn, because it's $19.95 and that gives you $5 off. And even if you don't like it for any reason at all, you can just call us up, write us, email us, whatever you want to do, and say, I don't like it, I want my money back. We give you your money back. That's what we mean by risk-free. Yeah. You have to plunk down your credit card or your debit card or whatever you use to buy it, because I find if you don't have skin in the game, you won't value it. You have to, get skin in the game up front. Buy it for $14.95, and if you don't like it, it's very, very easy. Just a little email or a telephone call. Tell us you want your money back, and you get your money back. Obviously, very few people do, or we wouldn't be in business for 15 years selling Sweetamine successfully. Try it. Doesn't cost you anything ultimately, unless if you want it. If you buy it, ultimately people usually buy it in quantity. Regular customers will buy 3, 4, 5, 6 a year's supply at one time. You buy it in bulk like that, it costs you less than $1 a day. So that's it's less than a cup of coffee.
Right.
It's very, very, very reasonable, and these packets are easy to carry around, travel with, and convenient, so you don't have to worry about bringing it along with you. Where's my satchel of Sweetamine? No, just bring a couple of packets of Sweetamine with you in your purse or whatever, and you can travel with it just fine.
Thank you for bringing nuance to the conversation and convenience to getting enough glycine. So Dr. Brind, it's always a pleasure. Everyone check out Sweetamine Autumn. Check out some bone broth. Just get your glycine. And thanks, Dr. Brind.
Thank you again for having me on.